on. Okay, so just as a metaphor of what we're trying to do, this is a schematic of the healthy auditory system. So you have the cochlea that many people have been talking about, inner hair cells, outer hair cells. The signals go up through a chain of cochlear nucleus and so on up to the cortex. And of course, there are lots of pathways going back down to the... So this is in the healthy system. So in the impaired system, this part here is damaged. And what we have available to alleviate this damage is the hearing aid. So that's something that's going to alleviate the... It's going to repair the pathway that goes up. But it can't restore the pathway going down. That part is missing. And so you could say that what we're trying to do is, in some sense, conceptually equivalent to restoring the efferent pathway that is lost in this case.
Alain?
Yes?
I think Tobias also alluded to the idea that the efferent pathway to the cochlea might play a role in selective attention. Is there really any evidence for that at all?
Of what we're trying to do. So, second part of my talk is actually doing the decoding part, where you take signals from the brain and acoustic stimuli. So, in auditory neuroscience, typically an experiment looks like this. You present a stimulus to the ears of the subject, and you record an electrical response from their scalp. And based on the knowledge of the stimulus and measurement of the response, you try to infer how perceptual mechanisms work. And there's, countless papers have been. So basically what you see is something that looks like this. So here's a time axis. Actually, it's a more or less logarithmic axis to compress all the different aspects of the waveform in a single graph. And what you see is basically a squiggle. A bunch of squiggles. And so based on the amplitudes and the positions of these squiggles, people have, there are thousands of papers making inferences on what is going on within the brain. So this is very, very nice. The problem is that in order to get these, because of the signal-to-noise ratio, the poor signal-to-noise ratio, you need to repeat the stimulus many times, and you have to average over repetitions, which means that you can't use, it won't work for continuous stimulation. For example, speech, it won't be very useful. So the stuff that we really care about, which is how we hear the world, you can't, there are limitations on how you can study it. So a new approach has been gaining momentum over the last few years, which is based on system identification or decoding. So basically you have an ongoing stimulus here, you record the brain response, and using the appropriate type of algorithms, you try to determine the properties of the system, usually as a linear system. And there are tons of papers that have come out. Mima Meskarani, Naiding, well, Jonathan Simon, who talked last week, is one of the major players, and so on. So there are essentially two flavors to this kind of work. One is based on a forward model, and the other is based on a backward model. So the forward model, you take the stimulus, and you try to find a linear model that is going to predict the EEG. With the backward model, which is also called stimulus reconstruction, you go the other way. You take the EEG, you find a linear model that can post-dict the stimulus, or reconstruct, quote unquote, the stimulus. So forward models, you take the audio. It's not really the audio, you have to derive features first. Like, it could be just the waveform envelope, or it could be a spectrogram representation, or it could potentially be something more sophisticated. And you predict the EEG, and it looks like this. So here, for example, if your feature is the acoustic envelope, you're going to fit a model like this. So you express the EEG at time t as a weighted sum of samples of this envelope in the past. And the solution to this gives you an FIR filter. If you're working with a spectrogram, it's exactly the same thing, except that your filter here is going to be a multi-channel FIR filter. Yeah. So you're expressing each channel. So here, you're expressing each channel of the EEG as a weighted sum of different channels of your spectrogram. So different frequency bands of your frequency channels of your spectrogram at different points in the past. So this is the sort of result that you get. So the model that you find can be described by this thing here. So it's they call a temporal response function, or a spectrotemporal response function. The S is hidden. And the prediction that you get looks like this. So if you're a believer, you'll agree with me that the red thing looks, well, the blue thing looks like the red thing. So the red is the EEG, and the blue is the best approximation, best prediction that you can make based on the stimulus of what this guy looks like.